So my name is Garm. I'm an associate professor at the University of Copenhagen. I'm a neurobiologist and I work on different neurobiological aspects. But one of the things that really interests me is how much processing can you do with a simple brain? The Caribbean species we're working on is called Tripedalus. It's a box jellyfish, and it's one of the smaller box jellyfish. It's about a centimeter in diameter, in bell diameter when fully grown, and then it has about 2 to 3 centimeters of tentacles. The big box jellyfish are 30 centimeters in diameter and have several meters of tentacle. So it's a rather small one. And it's not as venomous as many box jellyfish. They are renowned for killing humans in Australia. But this one here has pretty mild venom. It feeds on copepods. And this it does in between the prop roots in the mangrove swamps where they live. And here they are during daytime. And during nighttime, they rest on the bottom of the mangrove lagoon and wait for dawn to come again. Box jellyfish belongs to the animal phylum called cnidarians, and they are the earliest animals that have a nervous system. So what we can look at here is some of the earliest properties of nervous systems. And it's quite a surprise that already the first nervous systems, as our study indicates, would have the ability not only to learn, because that's maybe not so surprising, because simple forms of learning has been shown before. But this more advanced type, which we call associative learning or operant conditioning, has not been shown in these rather simple animals before. So that is kind of a surprise. It's also surprising when you look at the way the nervous system is organized in these animals, because previously it was sort of assumed that you needed one central brain for an organism to learn. And these animals don't have that. They do have a central nervous system, though, but it's very differently organized, not in one single brain, but more spread out throughout the animal. It has been the best of both worlds. We would have conducted these experiments in the wild, in their natural habitat. But unfortunately, there are too many parameters there we can't control. So what we did was to sort of fabricate a mimic of their habitat in the lab. And that's what the behavioral arena is all about. So we know that they use vision because these jellyfish actually have image-forming eyes. So we know they use vision to see the roots which they need to avoid. So basically what we needed to do in the tank was not to have actual roots in there, which would also add a lot of chemical and mechanical cues, but just to paint mimics of the roots on the side. So that was what we did. So they would be in the center of the arena, and then the edges would be mimics of the roots. And what we can then do there, without actually changing the distance to the roots, we can change their contrast. And the interesting part here is that the animals need to know the distance to the root. They need to turn before bumping into the roots, so they need to know how far away is the root. And for this, they actually use the contrast. You can say, how dark is the root relative to the bright water around it? And in a natural habitat, you would have a very dark root, high contrast close to the animal, and a low contrast further away. But what we, of course, using paint could trick the animals by is painting a low contrast root that would be close to the animals. And this was what we did. And then we observed how did the animal then behave when they were tricked in this way. Yes, initially, because we tricked them, they would bump into the roots. And that means that they get this mechanical stress signal by basically having a hard bump into this painted root. And already after 3 to 6 areas trials where they did this bumping into it, they learned that today apparently low contrast mean close by. And then they learned to turn earlier and thereby avoid the roots. And that is what we call associate learning or operant conditioning, that they're able to combine two different stimuli, what they see and what they feel, and correlate these two inputs. And thereby, through the visual input, predict that when they see a thing, they would expect a stress signal in a minute if they don't turn. So they learn to predict this and thereby change the future by changing their behavior. Many things. And as I tend to believe it often is, it's other scientists. It's basically meeting the right people at the right time in your life And that's what has been inspiring me. The first big inspiration throughout my career was my PhD advisor, another Danian, he was called. And he's working on crustaceans. So that's why I started out. And very soon I, along with Jens, discovered how much interesting work can be done on sensory systems and nervous systems. And back then it was olfaction. And then during my PhD, I ran into sort of my next supervisor. And then I moved to Sweden for my postdoc career with Dan Nilsson in the Vision Group in Lund. And that was when I changed my research topic more to vision and visual processing, as this study is also about. And I think Dan Nilsson is probably the biggest inspiration throughout my scientific career.